A-Level Biology 9700, Paper 4, Variant 2, May June 2025, question number 6. And question number 6 is guard cells are located in the epidermis of the leaves of most plants when animal environmental conditions change. This causes changes in guard cells that control the opening and closing of stomata. Describe the structure of the guard cells. So here we have to describe the structure of guard cells. And what are guard cells? The guard cells are the are specialized cells which are present in the epidermis, which is the outer layer of the leaf. So the guard cells are present in the epidermis and especially the lower epidermis of the leaf. So their primary function is to control the opening and closing of stomata. Stomatas are the pores which are produced between the guard cells when they are open. So the exchange of gases takes place through that guard cells. So here we have to describe the structure. If first of all, if we discuss the shape of the guard cells, they are typically found in pairs and are often kidney bean shaped, in dicots or dumbbell shaped in grasses and monocots. And if we discuss the cell wall is unevenly thickened. The wall facing the stomatal pore is generally thicker and less elastic, while the wall facing the epidermal is thinner and more elastic. And cellulose microfibers are also there, readily oriented cellulose microfibers present, radiating out from the pores. So this specific arrangement prevents the cell from expanding in diameter and instead forces them to curve outward when they become turgid, thus opening the stomata. So chloroplasts are also there because the process of photosynthesis takes place in these parts of the guard cells. So guard cells contain them and the process takes place. So vacuole is also present. They possess a large central vacuole. They play a crucial role in regulating turgor pressure through the movement of the water ions and nucleus. Proper nucleus is there, just like uh, in any other eukaryotic cells. And then you can see these mitochondria are present because they have to perform the process of photosynthesis. So these are you can say the different parts of the guard cells. So first of all, I would like to start with the first part, that is we have to start with the shape of the guard cells. They are typically bean shaped. And we can add a word kidney bean shaped and exist in pairs. The cell wall is unevenly thickened. Unevenly thickened. The wall facing the pore is generally thicker and less elastic. Then the other part, on other side. Cellulose, microfibrils are present in cell wall, radiating from pore due to which they are curved in shape. Chloroplasts are present unlike the epidermal cells. For photosynthesis. Large central vacuole and PS is also present as in other eukaryotic cells. Mitochondria help to perform the function of photosynthesis by providing energy. So it was all about the structure and part A of this question. Now we are moving towards the next question. And next question you can see is a graph. And what is in graph we will discuss. So question is, stomata have daily rhythms of opening and closing. Figure 6.1 shows the percentage of stomata opening at different times of the day over a period of 3 days in the tail crest branch, Arabidopsis and Thaliana. So this is the plant which has been shown with the help of this graph, the percentage of stomata opening and closing. So the 12 midday shows 24 hours shows the midnight, and this black line shows the black thick line shows the darkness. So here you can see that at 6 hours, the percentage of stomata open is 90%, and then at 12 hours it is 93%, and then you can see at between the 12 and 24 hours, that is we can say that is 18 hours. So at 18 hours it can be this 80%. So it is 80%. So in this way, if we see or if we observe in the day one, this is maximum 90%. So this is maximum 90%. And the other day you can see at 6, it is 80%. And then at 12, it is 70%. And then at 18th hour, it is just like this. You can see it is less than 30%. So same is on the third day. You can see at 6, it is 63%. This is 63, and then the maximum is 90%, and then it is dropping down. At 18 hours, it is less than 40%. So in this way, all the 3 days, the darkness, the black bar which spans from 18 to 6, or the portion of the 24 to 24, the percentage of open stomata drops significantly to its lowest point indicated stomata clear. So the minimum percentage of open stomata falls to around 10% to 20% or shortly after. So this pattern is repeated over the 3 days, demonstrating a cyclical and regulated process which is often controlled by the plant's internal biological clock known as the circadian rhythm. So the opening in the day allow for carbon dioxide uptake for photosynthesis, while the closing at night minimizes water loss through the transpiration. So now we can write all this observation in the space given, that is. So with the reference to figure 6.1, describe the rhythm of stomata opening and closing. So here I have described something about this graph. So it is only of 2 marks. So I would like to say that we have to write very briefly the answer. So first of all, I will write in this way, that is the day 1. We can see 90% are open, and in day 2 maximum they are 80%, and in day 3 they are again 90% open. So the maximum opening is about 90%. So after you can see the different days, they are changed. So 90% and at midday, and with the observation, I would like to say the during the marked, you can say the period, that is the which is mentioned with uh, black thick line, which spans for approximately you can see 18 hours to 6 hours or 24 hours to 24 hours. So the percentage of open stomata drops significantly to its lowest point, indicating stomata clear. So the minimum percentage of open falls to around 10 to 20%. So after this, when the daytime is there, it is open. So during these days, I would like to say the pattern is opening and closing. So this is the pattern. Opening and closing, this is the pattern. The first the opening, then closing. So this pattern is repeated. So pattern is repeated over the 3 days. Over the 3 days, demonstrating cyclical and regulated process. The opening in the days allows for carbon dioxide uptake for photosynthesis while closing at night minimizes the water loss through transpiration. So this is the whole process. In the daytime, when there is light, the process of photosynthesis is taking place. The opening and of these stomatas is about average, we can say the 90% on the day one, and on day 80% on the day three, it is 90% again. So the if we see the mean value, so it is between 80 to 90%. And when the day is passing on, and in the evening, the percentage of opening is decreasing, and when there is no light at 24 hours, there is stomatas are completely closed, which has been shown that there is darkness and no process of photosynthesis taking place at that time. So this is the cyclic process which taking place between the 24 hours. Now the next question is, suggest other environmental factors apart from the time of the day that can contribute to this stomatal layers. There are many other environmental factors due to which the process, you can see, of opening and closing can be stopped. So here, first thing is then when the soil is dry and the plant experience water deficit, the plant releases the hormone with cyclic acid. So signals the guard cells to lose to stomata to cause a closed to produce water loss through the transpiration. And then when there is high temperature, extreme height can increase the rate of transpiration to unsustainable levels. To prevent excessive water loss and potential damage, the stomata will close. So high concentration of carbon dioxide, if the concentration of carbon dioxide inside the leaf is air species becomes very high, the production from transpiration exceeds the rate of use in photosynthesis. So the guard cells respond by closing the stomata. So this is regulatory mechanism high when there is strong winds can rapidly strip away the humid boundary layer of the air surrounding the leaf, so increasing the rate of water loss and often leading to stomatal closure as a drought avoidance mechanism. So I would like to write on the two. So I can write first of all the when there is high temperature can increase the rate of transpiration to prevent excessive loss stomatas will close. Secondly, secondly, when there will be stomatas will be closed, and secondly, when there is high wind speed, which can rapidly strip away the human boundary layer and it will lead to stomatic. So there are many different factors due to which the stomatas can close. So it was all about this question. Now moving towards the next question. And next question is, the table shows some of the events occurring during the closure of stomata. The events are not listed in the correct order. So the table shows, so water levels the guard cells by osmosis. So they have given one of them. They have given one of them. So they have given number 1 and they have given number 5 and 10, D, F, and C. So it means it will start from the D. It will start from the D, and D is plant is subjected to a change in environmental conditions. So it will be at 1, and then there will be after this, as a result of this, the plant releases, you can say, uh, when there is change in the environmental condition, this is the initial stimulus. It can be drought, it can be high temperature that can triggers. So second, What will happen as a result of this? The plant releases abscisic acid. So E is at number 2 point. And then after this, what will happen? The abscisic acid binds to receptors on the cell surface membrane of the guard cell, because when it will be released, so it will bind itself on the cell surface membrane of the guard cells, travels and bind to the specific receptor, so initiating the signals cascade for closure of these cells. And then after the number 3, then we will reach to number 4, which is so after this is the J, and number 3 and 4. It will be after this we will move towards the yes. After this it will be the 4. Active transport of hydrogen ions out of the guard cells stops, because at this time, the abscisic acid binds to receptors on the dead surface of membrane of the guard cells. So as a result, the active transport of hydrogen ions out of the guard cells will stops, because this will stop inhibits the proton pumps that normally pumping the hydrogen ions are out, maintaining the negative membrane potential and facilitating the potassium uptake. So then we will reach to the point 5, and which they have given. Calcium ions move into the, as a result, when there will be the higher concentration of hydrogen ions, so what will happen? The calcium ions move into the cytoplasm of the guard cells, and um, so calcium ions move into the cytoplasm of the guard cells, change the membrane potential, and its viscous signal causes an influx of calcium ions, which act as a key second messenger in the closing pathway. So after this, the potassium ions leave the guard cells, because there will be the higher concentration at that time. So they will move out. The potassium ions will leave. And then what will happen at 7? The water potential of the guard cells increases due to the influx of these calcium ions. There will be the water potential of the guard cells will increase. So then after this, when the water potential of the guard cells will increase, then what will happen? The water leaves the guard cells by osmosis, because water moves from its higher water potential towards the lower water potential. So it will move. So surrounding epidermal cells, it will move in the surrounding epidermal cells. Water moves out of the guard cells, and then guard cells become, when the water will move out, the guard cells will become flaccid. They will become flaccid. And then what will be there? They have given, that is the stomata close. So these are the which we have to write over here. D E G E J I A H and C. So I have described all in this way. So it was all about this question. Uh, Please like, share, and subscribe my channel for more questions and more papers, more solved papers for your exams. Thanks for watching.